Assalamualaikum warahmatullahi ta'ala wabarakatuh. Myself, Muhammad Sahal Abdullah from Marcus Unani Medical College, third year student. My topic is genetic variations and evolutions of novel coronavirus. So, as we know, coronavirus disease is the severe acute respiratory disease in human that is caused by the emergence of a new type of coronavirus. The outbreak of the disease was first identified in Wuhan, China in December 2019. And Wuhan city became an epicenter of the disease, and then the outbreak spread outside of the China and caused the emergence of the disease in 213 countries in all over the world. The total number of infected cases are, as we know, about to grow, and the death is about 8.5 lakhs. And it has been recorded globally until September 1, 2020. The World Health Organization, that is WHO, called it is a public health emergency of worldwide concern. So, the coronavirus family is an enveloped virus containing a single strand RNA, and the diameter of the virus is about 80 to 120 nanometer. Coronaviruses are divided into 4 types: alpha coronavirus, beta coronavirus, gamma coronavirus, and beta coronavirus. Some common human coronavirus, as we know, HKU1, NL663, and OC432298. Infect humans and only cause mild respiratory diseases, while MERS coronavirus and SARS coronavirus 1 and SARS coronavirus 2 cause deviously severe acute respiratory infections. All coronavirus in 2019 coronavirus, that's SARS coronavirus 2 with MERS coronavirus 1 and SARS coronavirus 1 belong to the same group of coronavirus. Beta coronavirus, the genome sequence homology between SARS coronavirus and the other virus are noted. So, both SARS coronavirus 2 and SARS coronavirus 1 is exploit the same receptor for the binding process, angiotensin converting enzyme 2 receptor. And this means that both viruses may share structure similarly and a way of attaching to the cell receptor. SARS infection, it has been found by structural model analysis that the new coronavirus, SARS coronavirus 2 affinity of binding ACE2 10 times stronger than the previously known as SARS coronavirus 1. The service spike in glycoprotein helps viral entity into the host cells by homotrimers projection from the viral surfaces. The spike protein has 2 main domains: S1, which is responsible to bind the host cell's receptor, and the S2 is responsible for the fusion of both viral and host cellular membranes. The most genomic variation part in SARS coronavirus and SARS coronavirus 2 is the receptor binding domain in the spike protein, and some locations in this protein sequence might be related to the positive secretions. Due to the abundant variability in SARS coronavirus 2 isolates, many questions require an answer to understand whether their mutations have a role in the pathogenicity of SARS coronavirus 2. This is significant to understand the viral infections mechanism and the way to find drug and vaccine to protect people from the next stage of pandemic. So, we take a material and methods to identify the pathogenesis and to conclude which mechanism is happening in coronavirus in SARS coronavirus 1 and the SARS coronavirus 2. So, first isolate was used as a reference strain in the following mutations. Multiple sequence alignment of sequences were performing using T-copy program. All amino acid viral sequences of the spike, nuclear plasmid membrane and envelope proteins were screened in both animal and human viruses, and the frequency of mutation was calculated statistically comparing using the X-square test. Then the Benjamin-Hosberg method was used to identify the results that were statistically significant in multiple hypothesis testing. A strong false discovery rate of 0.001 was used to determine the statistical significance. So the result was the pandemic COVID-19 is the third known outbreak to cause respiratory illness in the current century. It has been caused the zoonotic coronaviruses, and it has thought that jumped from the bats or the pangolin host to humans. The disease spread mostly everywhere globally very rapidly due to traveling. Severe mutations have been observed in the virus genome during the rapid disseminations of the disease across different continents. Antigenic drift may occur as a result of the viruses to survive due to natural selection process, like it is common among influenza viruses. So I am concluding the topic. The different variants of coronavirus have been identified infecting humans and animals. The differences in genome structure and the sequences make the coronavirus to be different severity of infection and host selectivity. Coronavirus continuously change their structure by mutation, deletion, and insertion mutations. And through these three processes, that is mutation, deletion, and insertion, the coronavirus is mutating and making variables. So coronavirus continuously change their structures. First, SARS coronavirus 1 and SARS coronavirus 2 is the RBD in the S protein, and some locations in S protein sequence might be related to the positive selection. Due to several changes in the genome sequence SARS coronavirus 2 isolates, it is necessary to find the location of mutations and understand the role of these mutations in the pathogenicity of SARS coronavirus 2. This is significant to understand the viral infection mechanism and where to find drug to protect people from the next stage of the pandemic. So I am concluding, as I tell, we want to find the mechanism how the virus is acting in human body and how the virus is acting in the animal body to find a drug to prevent the coronavirus. So I am concluding. Assalamu alaikum warahmatullahi ta'ala wabarakatuh.